Okay, everybody, I'm back for translation. Okay, so this is part 2 of our lesson. Translation occurs also in the exact same 3 stages. So we've got initiation. What we're looking for here is rRNA, MET tRNA are going to join with mRNA at the start codon. Okay, so that doesn't make a lot of sense yet. It'll make a lot more sense later. I'm going to show you what a tRNA looks like. For the time being, know that R stands for ribosomal, okay, and T stands for transfer. So we have 3 types of RNA. We have mRNA, messenger. We've got rRNA, ribosomal RNA, and we've got tRNA, which stands for transfer RNA. rRNA is simply a ribosomal unit. So I'm going to show you that, but we've always told you, since you've been learning about organelles, that ribosomes are responsible for making proteins. Today you're going to finally find out why that's the case, okay? And then elongation means we're just going to make our protein. Not really our protein, but our polypeptide, okay? So we're going to find amino acids that are coded for on that mRNA molecule, and we're going to assemble peptide bonds. And then when we reach a stop codon, we're done. That's termination. Okay, so you're going to see all that unfold in the next few minutes. So what I suggest is, if you don't have it in front of you, which you probably don't, pause the video and go get your genetic code. You're going to need it. Okay, going back here, we have the mRNA molecule that I made the other day, or a few minutes ago, with you guys. Okay, so this is our 5' end. This is our 3' end. Give yourself as much space as you can because it's going to get a little messy, okay? So first of all, we have initiation. So you have to say to yourself, what is the start codon? I've mentioned this start codon before. So I made you guys highlight it. It's AUG. That's our start codon, okay? The amino acid that AUG codes for is actually methionine. So we'll just call that MET, okay? And so MET, AUG, is our start codon. I'm going to highlight it. I might regret it later, but I want you guys to see that. Okay, that's your start codon. And so this is where initiation begins. All right? And so what's going to happen here, I'm just going to draw this down here. You're going to have a ribosomal unit. And so remember when you started organelles and I made you guys look at your organelles and make that summary sheet? I said to you, be aware that the ribosome has 2 subunits. Now I'm going to do this in pencil because I am very limited with artistic abilities. So we've got a small ribosomal unit, okay? And for lack of a better way of drawing it, I'm just drawing it like this. And I probably should have drawn it bigger. And the ribosomal unit, this is the small part, so 2 subunits to a ribosome. It has 3 sites on it. And the sites go in this order: E, P, A. Exit, polypeptyl, and aminoacyl, okay? That's something that you can look up in your books, all right? So I think that's in the textbook. If it's not, actually, I'll just write it down here. So we've got E for exit, P for polypeptidal, I think that's how it's spelled, and then A is aminoacyl. Okay, and these are your 3 sites on your ribosome. All right, a ribosome is made up of 2 subunits. This is the small one, and then we've got another one. And the way I like to describe it best, it's like a baseball glove is going underneath and catching that. Okay, so it's like a baseball glove can wrap underneath this and grip it. So we're going to have this other big ribosomal unit. Okay, and the textbook obviously does a much better job of this. And the large ribosomal unit also has an EPA site. Okay? So I happen to have this open for you guys. We're going to look at page 327. If you want to see a better diagram, and there are many better diagrams than that one of your rRNA. Okay? So we've got the exit site. Oh, there's the peptidyl and aminoacyl. Okay? So this here is your small ribosomal unit, and this one here is the small ribosomal unit and the large ribosomal unit. So they're showing it up above, okay, which is probably better than the way I've drawn it. Okay, so notice you've got a small unit, a large unit, and together what does this make? This makes a ribosome. We call it rRNA, but it's your ribosome. Ribosomes make proteins. All right? So that's on page 327, a far better diagram than mine. So the combination of the small ribosomal unit, the large ribosomal unit, finding the start codon, in addition to finding this last type of RNA, that's all going to be called initiation. So the last thing I need to show you is the fact that you're going to have, and it doesn't look like this, I will show you what it looks like, but most biologists will draw something like this. Okay, you're going to have this molecule called tRNA. Okay, so you're going to draw T with 3 prongs coming off. And this is tRNA. But I don't put your label up there because this has to carry something. Okay, so it's carrying something. You might already guess what it's carrying. And this is tRNA. So you label it somewhere. I'm not labeling it because it's going to get too messy for you guys to see anything. Now at one end, the tRNA is carrying an amino acid. Okay, and so we're going to get to that amino acid in a minute. At the other end, it has an anticodon. Okay, the anticodon is for this codon here. So every 3 base pairs makes a codon. So you can see I've already worked this out so that we're going to have 5 different codons we have to read. Obviously, if this was a real mRNA molecule, we would have way more than 5. So what we do then is we find the AUG, and this tRNA is carrying the anticodon to AUG. So it's going to be UAC. All right, and now why is it carrying the anticodon? Here's the reason, guys. Okay, this is important. It's because this can form hydrogen bonds. All right, so we're going to form our hydrogen bonds, and that means the tRNA is going to stick there. All right, if it was carrying the codon, it wouldn't stick. It wouldn't form hydrogen bonds. This makes this partnership much more stable. All right, and then I'll show you what a tRNA actually looks like. It's way better than this. And what you have to do now, this is really super important, is you have to read the codon. This is the DNA message. Do not read the anticodon, ever. Read the codon. And so the codon is AUG, and you look at your genetic code and you go, okay, AUG, this is methionine. And this one here is important because you should always get met at the beginning of translation, okay? So initiation happens when this gets together with our RNA. I'm drawing it as a big blob, okay? And another little blob here. Remember, there are EPA sites here. Might be the worst RNA I've ever drawn. Okay, so we're gonna have EPA sites on both EPA. Now, this is hard for me to do, okay? And I might set you up with another video so that you can see it actually happening. Initially, what would happen is that this AUG would be engulfed with the amino acyl site. So in other words, shift this over, okay? And in reality, I'd have an E site here, a P site here, and this would be the A site, okay? And then as soon as all this comes together, now we've finished initiation. And so what we're going to do now is we're going to elongate, okay? So we're going to go through elongation. What does that mean? It simply means that we're going to read this, okay? And so when that happens, initiation is done. This site, this would actually be occurring, this would be occurring in the A site. And so what that happens is that the ribosomal unit then moves down, okay? And so after that, we would have the tRNA in the peptidyl site, okay? So it would be in the P site, which would open up your A site, all right? So imagine at that point in time, this is the P site, this is the A site, and then we've still got an empty E site, okay? All right, now we read the next set of codons, all right? And what you're going to see there is you're going to see CCC, but never, and then you have to have your GGG here. Put your tRNA molecule in, okay? And tRNA is carrying an amino acid. I'm going to erase my P from that site. And you have to say to yourself, never read the anticodon, always read the codon. C gives you proline, okay? And so what you're going to do is put a pro in there. So this is a lot of fun. I personally have a good time with this because you're just solving a puzzle from here on in, okay? Yeah, there's a lot of biochemistry involved, but it's fun stuff. So once that's in there, okay, so you imagine right now we're looking at this really being the A site. So I'm just going to move this up a little bit. This is the A site. This is the P site. So amino acyl, in other words, here we're going to introduce our amino acids. P, okay, when you think of proteins, you think hopefully P for proteins, but you must also hopefully think of a peptide bond, okay? And so this is why you started with this in Unit 1. What happens in the P site is that we form a peptide bond, okay? So I will put that in a different color. And that's what's going to happen. You're going to form a peptide bond between met and pro. And so again, if we imagine this as EPA, once this gets into that site, we form the peptide bond. And now this whole ribosomal unit will shift over to what I've drawn, okay? And at that point, this is what happens. E stands for exit. What's going to happen is that this tRNA is going to exit, okay? So I get to do this because I'm on a video. Unfortunately, I've left some ink in there, but that tRNA is gone, all right? It doesn't matter because the amino acid is now bonded to this one via a peptide bond, okay? So this is a peptide bond. Keep that in mind. Label it on your notes, all right? The tRNA is gone. It's going to get another met. This would be gone as well, okay? So I can't get rid of that. Actually, I can. Why not? All right, and our tRNA has exited, and what's it going to do? It's going to find another met for the next set, okay, for the next time we have to use it. All right, now we're finally with this A site, and so another tRNA is going to come in, okay, and we're going to have an anticodon of AAA so that it can stick in there, and at the end of this tRNA is carrying an amino acid. You have to say what is UUU. And so UUU is phenylalanine, okay? And that's Phe. And so you write Phe in there. And now once that's done, everything shifts over. This forms its peptide bond. And the whole process just continues just like that, guys. Okay? And so because I am out of space and I'm not going to move this over, know that everything would shift over one and we'd now be over here. Okay, so just out of pure convenience, I'm going to show you this tRNA molecule without moving my ribosomal unit over. And we'd look and we'd say, okay, this is CGU, but we read this codon and we say, what is GCA? So GCA is alanine, all right? And you put an ALA in there, we'd form a peptide bond, and then we're on our last transcribed from transcription codon, and so we have AUU, all right? And AUU is our anticodon, all right? Serves the purpose of forming the hydrogen bonds to keep everything stable. Keep in mind this would be in the A site, this would be in the P site, and this would be in the E site at this point. And so what does UAA code for? UAA codes for stop, okay? And so I purposely obviously did that. And there is no amino acid associated with your 3 stop codons. So I tricked you and I put that there, but there is no amino acid. So it's not carrying anything, all right? And so what that means is that this is our polypeptide. This says terminate, okay? In other words, we have finished protein synthesis. We are terminated, okay? That's our point of termination. And so what did you make? You made met, pro, phe, ala. So in some way or form, I'm going to be evaluating you guys on your understanding of this, okay? Typically, obviously, I'd have like a quiz with lots of short answers. You'd write everything out, as you know. That's not going to be the way to go this semester. I'm going to think about that, and then everything will be posted again for Monday or Tuesday. Okay, so there's going to be a thinking assignment. it's going to be easy in the form of the fact that all you need to be able to give me is mostly this. You won't have to draw everything out. Couple short answer questions, okay? And I'll get that put into the assignment box when I come up with it later today. And then now we're ready, after these 2 lessons, to have an LMS quiz. The LMS quiz is going to cover everything I've gone over with you, okay? So I'm going to make one more video taking up the homework. Maybe 2 videos are necessary because I know there's a bit of homework that I haven't taken up. We are done with protein synthesis. Awesome. I hope you understood it. I'm going to put a supplementary video up there as well from YouTube. I hope this quality is way better. I actually made a YouTube channel. I never thought in my whole life I would do that, but I know that the video quality was not good. I don't know if you were even watching the videos that I was making. Maybe just for the oral lesson you were watching them. I hope this makes you guys a lot less stressed, that you can actually see much better what I'm drawing. All right, and just before I finish, I do want to call your attention to a tRNA molecule. So if you look on page 325, this is what a tRNA molecule actually looks like. So it's really kind of cool, very complex, but what you have are nitrogenous bases, okay? So you've got your nitrogenous bases linked together. It loops out like this, okay? If you're at all interested, you can do some reading on here. There's a lot of interesting stuff about tRNA, condensing it this semester, so I'm not making you know that, but I do want you to be able to recognize it. So there's our amino acid that it's holding on to at the 3' end. It actually has a 3' end and it has a 5' end, okay? And then it loops down here and there's our actual anticodon, okay? And that anticodon is what would attach to the mRNA, all right? Isn't that awesome? Okay, I think it's pretty awesome. Now, I think that's all I need to tell you. So I hope you understood that, and I hope that you have a great weekend or had a great weekend, depending on when you're listening to this. All right? Take care, guys.